our project is to develop a new method that we've developed in the lab and apply it for epigenomic profiling to the goals of the 4D Nucleome project. And when you think about epigenomic profiling, of course, you're going to think of chromatin immunoprecipitation. So you might wonder, well, why do we need a new method? So let me just go through this. Even though chromatin immunoprecipitation is very familiar, the way it's generally practiced, it really hasn't changed for some 30 years, except for the last step, and that's the readout. So you start out by cross-linking cells and then shearing them, usually by sonication. You add an antibody for immunoprecipitation, and then you purify the DNA. And ChIP-seq has been out there for now 10 years, and there's some 55,000 articles on it. So it's one of the most widely used methods in all of molecular biology, but there's some limitations. So let me go through these. So first of all, you need a lot of cells. The efficiency is quite low. The background is high, and that's because the sonication basically pulverizes everything in the cell to solubilize it, and then you have what you're doing when you add the antibody immunoprecipitate is just to kind of pull the needle out of the haystack. So you're always going to sample from the rest of the genome, and that gives you background. The resolution is low in that the sonication means that your fragments are going to be hundreds of base pairs, and that doesn't take advantage of the single base pair resolution that's possible using a sequencing readout. And then I think this is kind of insidious. Most antibodies fail. For example, there was a paper that came out in Nature Methods last month, where this is a large NIH-funded consortium that made 1,400 mouse monoclonal antibodies to human transcription factors, and they were very high specificity based on protein binding microarrays, and they also were validated by immunoblotting or by immunoprecipitation. However, only 16% of a large sample of them actually passed using ENCODE criteria by a very experienced group. So most antibodies fail, and that means that that could be very expensive and doesn't take full advantage of the resources available by this multi-billion-dollar industry that makes antibodies for us. And then there are artifacts. I mean, everything can have artifacts, but the problem here is that there are various artifacts that people have discovered, such as hyperchippable regions and phantom sites, and so on. And the problem there is that if you only have one method like ChIP that people do, there's no true validation. So you're not really validating by doing qPCR on ChIP because the artifacts actually occur before the readout. So it's worth considering other methods. Now, there's a class of methods that I refer to as enzyme tethering. The first of these was developed by Boston and Steinsel almost 20 years ago, and that's DMID. With DMID, you make a fusion between your protein of interest and E. coli dimethylase, and the methylase will methylate the adenine of GATC sites in the vicinity, and then you map the sites. Now, a few years later, Uli Lemley introduced what he called chromatin endogenous cleavage, or ChIP-seq, by substituting eminase for DAM, and this gives base pair resolution that's possible by the cleavage by eminase, and also allows control using calcium. And Gabe Zentner, when he was a postdoc in the lab, developed this with a sequencing readout. But what I'm going to talk about is chromatin immunocleavage, which is what our method is built upon, or ChIP. Now, ChIP sounds so much like ChIP, people get confused about it, so we refer to this as cleavage under targets and release using nuclease, or cut and run. It goes like this. We mix live cells with lectin-coated magnetic beads. They stick. We permeabilize. At the same time, we add an antibody to diffuse in and find its target, say a transcription factor. Then we add a fusion protein between protein A and eminase. Protein A will bind to the antibody, will anchor the eminase, such that when we add calcium and their cleavages on both sides, the fragment gets released. We allow it to diffuse out into the supernatant, and then we extract the DNA and do paired-end sequencing. From live cells to purified DNA takes less than a day, and the backgrounds are extremely low. And that's for at least two reasons. First of all, when we add the calcium and activate, we worry about the diffusion of the particles will basically chew up all the accessible regions, and that would be an artifact for us. So we do it at a low temperature, and that limits diffusion. And secondly, unlike ChIP, where you're pulling needle out of a haystack, we leave the haystack behind on the beads. So here's some evidence that we get very low background. So in this case, we're looking at, we use an antibody that was used by the ENCODE project. It's a rabbit monoclonal for H3K27 trimethyl. We're doing this in human K562 cells. And if we sample the same number of reads for ENCODE and CUT&RUN, I think you can see we get much better feature definition with CUT&RUN. And we attribute that to the fact that the backgrounds are really quite high for ENCODE, so you have to sequence quite deeply, and that's expensive. So we think that CUT&RUN is an order of magnitude less expensive. And then we can go down to low cell numbers. So here we see in this experiment, we went down to 100 cells without any noticeable loss in data quality. So in contrast to the millions of cells, we can apply this to low cell number applications. And we get base pair resolution. So this is CTCF. And what we see is that whether we digest for 5 seconds or almost a half hour, and we map the ends, the fragment ends, so this is a footprint map, what you can basically see is that we get exactly the same 44 base pair footprint, regardless of the time of digestion, so it's pretty well behaved. And then if we look at a particular site in the genome, we see that we get a nice sharp cut-and-run peak, and we compare that with various ChIP methods. And I think if you look at the y-axis, we get much better at signal to noise. Now, I'm showing here just 120 the small fragments. If we look at the large fragments, interestingly for CTCF, we see that we release nucleosomes on either side of the CTCF binding site. Our interpretation of this is that the fiber flexes enough to allow the linker regions of neighboring nucleosomes to come close enough that they get cleaved, and that will release the nucleosome. And we realize that maybe that means that we could also be releasing contact sites from sites that come close to CTCF sites, and that turns out to be the case. So, for example, here we're comparing our CTCF cut-and-run data to ChIP-PET fragments. And for example, we have a fragment here shows this dumbbell showing connection of two restriction enzymes by a contact, and both of them have a cut-and-run signal. In fact, over 90% of the high-scoring ChIP-PET fragments all have cut-and-run signals. Furthermore, when we do native ChIP, which will only map the sites that are directly bound, won't map the contact sites because there's no cross-linking involved. You can see that this site here has CTCF, but this one apparently doesn't. And so that allows us to distinguish direct from indirect bound sites. And because Hi-C and related methods, basically they're about restriction fragments size, we get base pair resolution with cut-and-run. So I think this really enriches the kind of data that you're collecting when you do 3D contact mapping. Okay, so what are the various applications of this? So first of all, I'd like to point out that we can do salt fractionation. Now, salt fractionation is a classical method that goes back to the 70s, where low salt will extract chromatin after micrococcal nuclease digestion. Will extract chromatin that is so-called active chromatin, that's where the term came from, and high salt will give you the silent chromatin. So what we do is we extract the supernatant and low salt, and then we wash the bead nuclei with high salt, and we sequence that and the pellet. Now, where this has turned out to be especially useful is for human centromeres. Now, although this is the 4D Nucleome project, and you would think the centromere would be the most get the most attention in the project, because I mean, of all the nuclear bodies, this one is the one that's most important and most familiar, and yet there almost nothing we know very little about it, and that's because of the technical problem that centromeres are extremely homogeneous repeats, and we really haven't been able to map them even now. However, we've applied cut and run and gotten over at least one problem of studying centromeres, and that is that they're extremely insoluble. And that's allowed us to actually probe using cut and run the centromere chromatin complex. Another application is to turn cut and run into a diagnostic tool. So Jay Sarthy, and this is in collaboration with Derek Janssens, who's been developing cut and run as part of the 4D Nucleome project. Jay is a pediatric oncologist, and so in this particular experiment, from autopsies of patients, by dissecting, by basically removing the tumor from two different patients from the same part of the brain. So this is brain tumors. Then they get passed through mice, and the tumors grow in the mouse brain. So these are so-called patient-derived xenografts. He took two of them and also had cell lines. And these are flash frozen and then thawed. You see them here. Disaggregated, mixed with Con-A beads, arrayed them on a plate. And then Derek put the whole thing on a robot, and we did the entire cut and run reactions on the robot, going right through the addition of barcoded adapters, including the cleanup. He then took the plate off, did PCR, put it back on the robot for cleanup, did QC, and mixed up the 48 samples. Now, we then do paired and 25 by 25 sequencing on a 2-lane flow cell. So when we calculate our sequencing cost, it comes out to about $25 for about 5 to 10 million map reads. So what can you get for $25? Well, here's a region, so showing first of all, from the two different patients, you can compare replicates between the cell lines and between the cell lines and the actual flash frozen and thawed tumors. And really, as far as we can tell, the data quality is very similar. In addition, what you'll see is between the two patients, although they came from the same part of the brain, they're very different tumors. They show these striking differences. Now, this is a particularly interesting region of the genome. It's the site of the P16 gene, which is one of the most commonly mutated genes in cancer. But in pediatric cancers, there are very few mutations. And so therefore, it's important to have a method that can, I don't know if you can see this, that can actually give us high resolution to show basically show what's happening at the epigenome level. And I think you'll notice that right over the promoter of the oppositely oriented P16 and the P15 genes, what you see is that there's a nucleosome and it's got an active and silent mark. And we think that that may actually contribute, although we don't know, may actually contribute to the cancer phenotype. Okay, now, since our paper was published a little over a year ago, we've had many requests. We've sent out the material, protein A eminase, to now some 250 laboratories, and some of our early adopters, some of the papers are beginning to appear. So here's one from a group from University of Massachusetts Medical School, where they show that they can use cut and run and map transcription factors in single cells where they have basically in a 96-well format. I think this is very impressive to be able to get down to such low cell numbers. This speaks to the efficiency of the method. Then in a paper that appeared a few weeks ago in the journal Cell from Stuart Orkin, they applied cut and run to the BCL11A repressor. Now, BCL11A has been notorious for being resistant to ChIP-seq. Many groups, including Orkin's, have failed with it using many different antibodies. For Orkin, cut and run worked right away with different antibodies. The one that they actually used was one that actually wasn't recommended for ChIP in this study. So they first were able to get high-resolution mapping of BCL11A sites throughout the genome, and by running MEME, they were able to show that they could get a single motif, TGA-CCA, that precisely mapped what they were able to obtain using protein-binding microarrays. Furthermore, and most impressively in this paper, it turns out that there are two TGA-CCA sites in the gamma globin promoter, and that's what BCL11A binds and represses. If you lose repression from by BCL11A at the promoter, then what happens is that it will basically turn on the gamma gene. And reawakening the gamma gene has been a goal for decades. But it wasn't known exactly where the switch is. So the switch turns out to be the site. There are 2 sites in the promoter. They're only 21 base pairs apart, and the high resolution of CUT&RUN allowed them to map it to the distal site, and that turns out to be a site where there's a benign condition called hereditary persistence of fetal hemoglobin. That turns out to be that 6 base site is one that in individuals who have that benign condition actually is mutated, and they confirm that by editing it out. So they could use CUT&RUN in order to identify this important switch, and this leads the way to being able to reawaken the gamma gene by editing in individuals who have sickle cell anemia, because this would relieve, and beta thalassemia, because this will relieve the symptoms. I should also point out that they also took advantage of CUT&RUN to identify 3D contact sites between the enhancer in the LCR upstream and the promoter. So it was useful for that as well. Now, because we're so keen about using CUT&RUN for the 4D Nucleome project, we've been Giancarlo Bonera has been uploading our experimental data sets. And right now, actually, there are more CUT&RUN data sets, about a quarter of what you can get now in the 4D Nucleome portal. Actually, we have another set. This includes mostly K562 cells and also H1 cells, but we've also been using differentiated, differentiated cells, that is the definitive endoderm. And I just want to thank Dave Gilbert and the cells working group for helping us get this going in the lab. Now, these are not included in the 51 data sets available, but they're coming online now. We've already submitted them. And let me just tell you about that experiment, because I think it turns out to be pretty interesting. So here we put H1ES cells. We differentiated them in the lab and confirmed that they're differentiated. We have 11 antibodies in both cell types, and there are 2 replicates that run in parallel on the workstation. And we just do a one-size-fits-all. We just give the same time of digestion. So we weren't quite sure how well this would work. And Giancarlo has done a preliminary analysis on this. And what you can see is that, I don't know if you can see this, but it turns out all the active histone modifications are in one corner here. And then you have K27 trimethyl and PRC2 down here, and CT-CF in the middle. And let's take a closer look. So if we look at K27 trimethyl, what you can see is that the replicates are very concordant, but there's huge differences between the H1 and the definitive endoderm cells. And we see the same thing for K4 ME12, where we basically, they are sort of mixed around. However, we get the distinction between the two, and there's nice overlap. This is K4 ME3, and the up top is H2AX. Whereas in contrast, CT-CF, which is constitutive, we don't see any distinction between the 2 cell types. And then there's something IgG, that's our negative control, and something called NPAT. Keep that in mind, because I'm going to get back to NPAT. So if we look at a screenshot, and you're seeing everything that we did here, we haven't done any real quality control filtering. I don't know if any is necessary. And I think you can see that for H3K4 trimethyl and H3K27 trimethyl CT-CF, there's good concordance between the H1 and DE cells. However, there's a difference here. Actually, Giancarlo picked this out. If we take a closer look at this, I don't know, on my screen there's something blocking, but anyway, this is a particular gene here that's called PALD1, and I don't know if you can see this. Let me do that. Let me just move this aside, make sure you can see this. So what you're seeing here is that there's an active mark over the PALD1 promoter in H1 cells, silent in the DE cell, silent mark, whereas this next promoter downstream, we don't see any difference. PALD1 is the catalytic subunit of DNA polymerase delta, which is the polymerase that replicates the lagging strand synthesis. And so, as you might expect, it's active in the pluripotent cells and completely inactive, should be inactive in differentiated cells. And this turns out to be a quite striking difference. Now let me get back to NPAT. Now, Carla gave a great introduction to nuclear bodies. Now, Hall bodies are not the only nuclear bodies that are held together by coilin, and perhaps some of those spots that you saw in her slides were actually histone locus nuclear bodies. And it turns out that the reason that we didn't see it looked like it was a failed experiment was that the vast majority of the genome doesn't have any of this because they're just located over the histone loci. These histone loci are extremely highly expressed, and what we can see is we actually get a pattern from NPAT over the histone loci. So this also, as Carla showed for the Hall bodies, we can see, but here at very high resolution, we can see that each of the different histone genes in this cluster is actually bound by NPAT. Okay, I'm just going to close here and ask for questions. I just want to acknowledge Derek Hansens, who did most of this, Terry Bryson, who's responsible for our participation in the cells working group, Christine Kadamo, who makes the libraries and runs the robot, Georgia, my wife Georgia, who did the initial analysis, and John Carlo in Bill Noble's group at the University of Washington, who's been doing the analysis and the uploading, etc. So I'll stop there and take questions. Oh, and if you want cut and run, if you want to try it yourself, just send me an aliquot, and we'll send me your info, and we'll send you an aliquot.
Hi, Steve. This is